Okay, first things first, we use radiation therapy in the cancer model to break the DNA. In breaking the DNA, this triggers protein kinase activation and phosphorylation of the p53 protein. In doing this, we remove the MDM2 protein from p53. Normally, without DNA damage, the p53 stays bound to the MDM2 and is degraded by the proteasome. But in this case, since we do have DNA damage, we are going to have the p53 protein dissociate from the MDM2 after it's been activated and phosphorylated by kinase. So what we see later on is that the activated p53 now stimulates transcription of CDK regulatory proteins like P21 and many others. These regulatory proteins bind CDKs and stop the cell cycle. As we see here, P21 is created after transcription and translation of the gene, and that then binds to the CDKs and inhibits cell cycle activity. This is an example of many cell cycle checkpoints used to check a cell's viability to go into mitosis. Lastly, we notice that the apoptosis is also triggered by p53 activation. So this is a big deal because apoptosis is needed so that cells do not undergo cancer, as it is necessary for cells to die so we don't get tumor growth.